Hi, my name is Rajan Sawhney. I'm a computer science PhD student at the University of Hawaii, and today I'll be presenting on my research on better capturing viral biology using protein language models. I'll briefly talk about PLMs, their applications, and their limitations, especially when dealing with microbes, how we modify these PLMs to address these limitations, and demonstrate the improved capabilities of our models. PLMs trained on evolutionary scale data have learned the language and underlying grammatical rules of proteins in the form of high-dimensional vector representations, also known as embeddings. These embeddings are advancing proteomics, facilitating highly accurate structure and function prediction, evolutionary studies, and protein design. But these general purpose or foundational PLMs come with their own limitations. They do not perform well on taxa that are underrepresented in most protein databases used for training PLMs. And viruses are particularly affected. So, how do we address this limitation? By further training or fine tuning these models on domain specific datasets. Here we describe our fine tuning recipe. We started with three widely used protein language models, trained them on about 300,000 viral protein sequences, applied three different learning objectives, and instead of training the entire model, we only trained a fraction of the model parameters. Here is a depiction of our fine-tuning process. During training, for each pre-trained PLM, we freeze their original weights. Using viral protein sequences from VogDB, we then update selective model parameters, shown in orange, using a technique called low-rank adaptation. And these parameters are updated through three separate learning frameworks. First, masked language modeling, where the model has to predict masked amino acids. Second, classification, where the model has to predict which group the protein belongs to. And lastly, contrastive learning, where the model learns similarities between proteins from the same group and dissimilarities between proteins from different groups. After training, original weights and newly learned weights are added together separately, making use of previously learned information. At the end, we have three pre-trained models, 9 fine-tuned models, so a total of 12 models. To evaluate whether fine-tuning improves PLM performance, we conduct three main experiments using PLM embeddings. For the first experiment, we measured pairwise cosine similarity between sequences from the same group, amino acids at conserved positions, and amino acids at non-conserved MSA positions. Across 30,000 pairwise sequence comparisons and about a million residue comparisons, what was noticeable was that amongst ESM2 models, the contrastive variant in green showed distribution shifts in the right direction. For ProtTrans, for non-conserved sites, the median was higher for fine-tuned variants compared to pre-trained. And ProGen2 consistently showed high medians, indicating fine-tuning didn't change much for this model. For clustering, we conducted two experiments. First, using our test dataset, we clustered protein embeddings in high-dimensional space and evaluated generated clusters using silhouette score, a metric used to measure tightness within clusters and separation amongst clusters. Over 1,000 iterations of clustering randomly selected sequences based on silhouette scores, we observed that contrastive ESM2 generated the best clusters. As a further analysis, we clustered DNA helicase embeddings in two-dimensional space and found that our contrastive fine-tuned ESM2 not only separated helicases by families, which the pre-trained variant struggled to do, but it also differentiated functional subgroups within the GP4 family based on the strict presence or absence of the zinc binding domain, as seen in yellow clusters in image B, a distinction missed by classical phylogenetic tree analysis, as seen in image C, yellow clade. For alignment-based experiments, first, using soft alignments technique and embedding-based global alignment algorithm, we generated 30,000 pairwise alignments and used the number of aligned residues as a metric to evaluate each model's performance. Here too, we saw that contrastive ESM2 led to the most number of aligned residues. Next, we generated MSAs using embeddings through the vector clustering MSA algorithm and evaluated the MSAs using T-Coffey's transitive consistency score. Over 100 generated PCMSAs per model. Once again, contrastive ESM2 performed the best, with contrastive ProtTrans finishing second. We also generated a VCMSA for bacteriophage proteins and observed that contrastive ESM2 generated the MSA with higher consistency, occupancy, and mutual information, reducing pronounced gaps found in the MSA generated by the pre-trained model. In conclusion, we evaluated 12 protein language models on their ability to capture viral biology. We found that selectively fine-tuning models on viral proteins using classification or contrastive learning helps improve performance. Our fine-tuned models demonstrated clearer clustering of viral groups, families, and functional domains, while also producing higher quality MSAs with improved accuracy and fidelity. By accurately modeling viral proteins, our approach advances tools for better understanding viral biology and harnessing it for biotechnological applications. I'd like to thank NSF for supporting this work, my collaborators at SAIL and at the University of Delaware. Thank you so very much for listening.